Next lecture. Okay, hi everyone. My name is Sevinch. I'm going to be taking over from Mark for a while. So we're going to co-teach this class. So we're going to have it about half and half. And I have my office hours on Fridays at 3 p.m. Or by appointment. I'm pretty good with emails. So if you shoot me an email, I'll reply you back within a day for sure. If not, in 10 seconds. All right, so here's the schedule. So what we're going to do is we're going to talk about genomes and gene expression in the next 5 weeks or so with me. And then Mark Siegel is going to take over for molecular genetic analysis. And then we're going to switch around a little bit. All right, this is a question that I'm sure I'm going to get very soon. So I'm going to preempt and show you what you can do to do better at molecular cell, especially with the midterm coming up on Friday. So I'm sure that Mark Siegel has already told you to keep up with the coursework, because one of the reasons why you want to do it is you learn better if you actually recall what you learned, right? So this is the typical curve of a student working for a molecular cell. So they do nothing for a while, right? And then they study right before the midterm. And right after the midterm, they'll forget everything and then study again. And that's not going to be very good for learning, because what you learned in the past lectures are going to actually help you in the upcoming lectures. So this course is going to move forward and proceed. It's going to get a little bit more complicated, and you're going to need the knowledge that you had to continue. Okay, so keep up with the coursework. That means do your study questions, go to your recitations, go to the lectures, and keep up with your reading. All right. The second thing that I'm going to suggest, I'm going to dare you to turn off your laptops, okay? If you want. The reason for that is it's been shown over and over again, the students that actually keep their devices on during the lectures tend to do worse because they're distracted, okay? So even though you think you're the master of multitasking, there's a very likely chance that you actually are not so as good as you think you are. This decision is yours, but this is scientifically shown over and over again that it's not very good for learning. Okay, so if you dare to unplug, put away your stuff, pick a piece of paper and actually take notes by hand, right? Maybe you'll do also better in the class. All right, so now the science. All right, so what we're going to talk about in this lecture is basically what is in our genome. And you already had a description of the genome, which is basically an organism's complete set of DNA containing all the information needed to build and maintain that organism. And I'm going to get back to this information over and over again, because what you can think of as DNA is basically the molecule that is storing the information in your cell. So it has all the information necessary to make all the proteins and the functional products at the right time and the right place, etc. So some of the questions that we molecular biologists ask and genomicists ask is, how is this information organized? How is it propagated? So propagation would be replication, right, and the division. How is it interpreted, right? We're going to talk a lot about transcription, because that's the way that this information is interpreted. And then we're going to talk just a bit about what goes wrong during disease, because the disease happens if one of these processes actually don't occur normally, okay? So if the DNA is capable of storing information, then it should be capable of storing any information, right? And a few scientists actually were thinking about this like 10, 15 years ago now, and they said, can we actually code a computer program into the DNA and then read it back and see if we can actually read this program? And they were capable of doing this. So this was a highlight in 2017. It's a paper from 2017, but some proof of principles has been done before. So what they did is they took a book, a movie, and a couple of other things, And then they turned that information into a computer code. And then they coded that computer code into the DNA using the string of ATGCs. Okay? Then they took that sequence and sent it to a company to synthesize the DNA. So they take back the DNA. So that information is in the DNA, right? And they took modern technologies to read the sequence back and see if they could reconstruct the book. And they were capable of doing this. And in the next step, another group, what they did is they not only actually coded the information into the DNA, they actually cloned that piece of DNA into bacteria, let the bacteria duplicate and propagate that information, took back the DNA, sequenced the DNA, and reconstructed the information. So what they did is, this was the movie that they encoded into the plasmid DNA, in the bacterial DNA, and then this was the movie when they actually read it back. So it is pretty amazing that you can actually use DNA just as tape or a hard drive to actually encode information, right? So DNA is pretty good in storing and propagating data. The reason why people don't do that right now, instead of using tapes and hard drives, etc., is it's very expensive. So if that changes in the future, maybe that's what we will be doing, right? Pretty cool. All right. So if DNA contains information, you would imagine that the more complex organisms that are going to require more information to make them will have bigger genome sizes, right? And roughly that's true. So in like small bacteria, etc., have smaller genomes, and larger animals like mammals or flowering plants have bigger genomes. But this is a rough positive correlation. All right. So the human genome is organized like many vertebrate genomes into chromosomes. So you can think of this like if it's a book, it's like the chapters of the book, right? So it has about 3.3 billion bases. It's organized into 22 autosomes. Autosomes are every chromosome that is not a sex chromosome. And you have the sex chromosome X and Y. And humans are diploid animals, so we have 2 copies of each chromosome, totaling 46. And when I ask you, for example, what's on chromosomes, the first thing that you might say is genes, right? So genes are on our chromosomes. They are linearly arranged, so this goes one gene after another on a linear chromosome. So a gene is defined as the entire nucleic acid sequence that's necessary for the synthesis of a functional gene product. It could be a protein, a polypeptide, or it could be an RNA. So in humans, estimated gene number is about 20,000, 200, 500, something in between that. And where do we stand with that number of genes compared to other organisms? We stand between grapes and chickens. So we have just about the same number of genes, just like many other animals have. And we have more genes than some of the more simple organisms like bacteria and viruses, right? But it's pretty amazing that fruit fly also has just almost as many as genes as we do have, right? And yeast has about 6,000 genes. That's not too bad for a one-cell organism. And if you look at the genomes of eukaryotes that are smaller and larger, the reason why we have about the same number of genes compared to a eukaryote with a smaller genome is that smaller eukaryotes have more compact genomes. What I mean by that is they put many, many more genes in one linear chromosome than us. So if you look at a typical human gene, right? This is a human gene cluster. It has several genes here. So each of these green rectangles are an exon, and each of these little things are introns. And what you can see is that there's lots of space in between the genes, right? There's lots of space. There's some pseudogenes. These are genes that they look like genes, but they're not. They've accumulated too many mutations to be functional, and etc. But if you look at the yeast genome, which is much more compact, each of these green boxes are actually a gene. They're called open reading frames. They encode for one protein, one functional product. So you can see that their genomes are really packed while our genomes are kind of sparse with exons. And if you actually look at the human genome and categorize the different type of sequences that we have, what you will be struck with is that the coding regions of the genome, these are the sequences that encode for a protein. That's what coding means, right? Comprise about 1% of our genome. So 99% of our genome are dedicated to other things than actually coding for protein products. So if you combine all the introns and exons in the genome, it will make up about 25% of the genome. And so these are the protein coding genes, so they make up about 25%. There are about 25,000 of them in our genome. So the protein coding genes, they're organized in transcription units. A transcription unit is a segment of the genome that's transcribed, okay? And there are usually two kinds that we talk about. One is a simple transcription unit. And in a simple transcription unit, you've probably seen schemas like this many, many times. What you have is you have a transcription start site. You have a transcription ending site. You have a translated region that is the start codon and the stop codon. And right upstream of the transcription start site is the promoter sequences. And these contain sequences that are recognized by general transcription factors. There are also other regions of the DNA that is far from the promoter, but capable of binding to transcription factors that actually regulate the activity of this promoter. That means regulate whether a RNA polymerase II is going to come here and start transcription or not. And these regions are called enhancers, and we're going to talk more about them in transcription lectures. So this is a simple transcription unit. It makes one product. It has a single transcript. It does not have introns. In humans, we have more complex transcription units. What that means is that we have lots of different exons, and we have introns in between them. Okay? So we have the transcription start site, we have the translation start site, but the coding sequence is broken up into pieces. These are the exons that end up in the mature mRNA. So the typical exon size in humans is about 50 to 200 base pair. They're pretty short. And the introns are quite large, right? The median intron size is 3.3 kb, and the longest is 17 kb. So you can see that in our genes, we have like little snippets of the exons that are basically interspersed with very long introns. That's how we have the information organized. And 90% of the human genes approximately are alternatively spliced. So when you have your exons organized like this, and if certain exons can combine with each other, you can see that a single gene can encode for 3 or 4 or more different functional products, right? So that's a complex transcription unit. A single gene with introns has the capacity to encode for multiple different products. Any questions so far? All right. So we usually classify protein coding genes into two classes. One is called solitary genes. These are what you would find one copy per haploid genome. They account for 25-50% of protein coding genes. And then there's another class which are called the duplicated genes. And these are genes that have similar sequences nearby, and they are basically gene families. And you can find several to several hundred copies. So an example of a duplicated gene is the tubulin gene in humans, right? So how do you generate gene families? So these genes, like tubulin, they're very well conserved. You find these genes encoding for certain proteins, the sequence similar sequences in bacteria, in smaller eukaryotes, etc. But in humans and other mammals, you can find multiple copies of this gene. So what happens throughout evolution is that we imagine that there was an ancestral cell containing the tubulin gene and the sequence, right? And then this gene has duplicated in this ancestral cell, creating two different copies. So these two different copies will, over millions of years, going to accumulate differences in their sequences. So this is going to have a different sequence, and this is going to start having a different sequence. So their sequences are going to diverge. As their sequences diverge, their function also start diverging. So the alpha tubulin will start specializing for certain functions, and beta tubulin is going to start specializing for certain functions, right? And then so after this, if there is a speciation event so that you have the two species that are further diverging, what you will have is that the sequences of alpha and beta tubulin in species 1 is going to accumulate differences over time. That is independent, will be independently also accumulated in species 2 over time. So when you come to current time, right, you take mouse and humans, and you take our tubulin gene, and then look for genes in the mouse with a similar sequence, you're going to find the tubulin genes in the mouse. There are going to be some differences, but they will look similar, okay? And that similar genes, what we call homologous. They're similar. They're homologs, okay? There are two other terminology that you will use. So because these genes, alpha and beta tubulin, have duplicated and diverged from each other, these genes with similar sequences that are to each other within the same species are called paralogous. They're paralogs within the same species. And when we're talking about genes that have similar sequences and functions in between different species, these are called orthologs. Okay? So this is a gene family. They're all homologs. The genes that are similar to each other within a species are paralogs, and genes that are different are called orthologs. And if you take evolution or read about it, you're going to hear this terminology a lot. All right. So these were protein-coding genes. There are a lot of non-coding genes. Non-coding genes means that they encode, so they are transcribed to produce a functional RNA product. Okay? So these functional RNA products in humans, there are many, many different kinds of them. The first one that you will always think of will be ribosomal RNAs. These are long RNAs that are required for the structure of the ribosomes. You have tRNAs, they're microRNAs. These are little RNAs that regulate gene expression. There are about thousands of them in our genome. So these non-coding genes are not negligible. They're pretty cool things, and people have been identifying more and more of them lately. All right, so some of these non-coding genes, especially ribosomal RNAs, are transcribed from a locus that has many, many repeats of this. Okay, so these what we call tandemly repeated genes, like the ribosomal RNAs. And ribosomal RNA, you find hundreds of copies in the genome. So this is the rDNA locus, it's just a little cartoon of rDNA locus. So what you have is you have many, many repeats. So there are only 2 of them shown here. There are hundreds of these repeats in the genome at certain places in some chromosomes. And each of these repeats contains the 18S, 5.8S, and 28S RNA. Okay? In humans, there are about 400 copies spread into 5 regions. So each of these repeats are transcribed. Here's a question for you. So why do you think that we need many, many multiple copies of the rDNA in our genome, while many other genes like myosin or some receptors or any other type of genes like RNA polymerase II gene, they're found in single copies? But the rDNA, so that's encoding for the RNA products, like ribosomal RNAs, we find many, many copies. Can anyone have a guess?
Well, you can't use the same RNA for multiple things. Like if I have one mRNA, then you can like keep translating that one over and over again. But once you can't, the rRNA just goes into a ribosome. It's taken out. It can only be used once.
You can't use the RNA again and again. Yeah, it's a structural component. For one ribosome, you're gonna need one RNA, but you can use the same gene over and over again. You can keep transcribing the same gene, right? Mm-hmm.
Is it because it's needed in so many different places?
It is needed in so many more copies. So in embryogenesis, the cells are dividing every 24 hours, right? They're just crazy dividing so fast. And it has to replicate all this genome. It has to make all the proteins. It has to constantly copy itself. Like imagine you have to make one of yourself every 24 hours, right? You really need a lot of ribosomes. So there's about 5 to 10 million ribosomes per cell in embryonic cells, right? And if you have a single gene, you can only make so many ribosomal RNA. The reason for that is there is a speed to the RNA polymerase II, right? So that speed is about, let's say, 10 bases per second. And you can only pack so many RNA polymerases next to each other, right? So you can only produce a certain amount of RNA per second. And if you calculate that with a single copy, you're not going to go over about 10,000 ribosomal RNAs. And that's a lot less than 5 million that we would need, right? So to be able to meet the demand, what you do is you make many, many copies of the rDNA gene so that you can, in parallel, produce lots of ribosomal RNAs. Give me another gene that you might think might be expressed in many, many clusters, in many numbers. Another gene product that you might actually need in high demand. Mitochondria? Not necessarily. Mitochondria, you don't have millions of copies.
DNA polymerase?
Not really. You actually don't need that many DNA polymerase. Think of structural, right? What would you need?
Can it be like a type of cell, like a red blood cell?
That depends. I'm not sure about that. Tubulin, you do have a few copies, but you actually, you don't need to make that much of it.
tRNA?
tRNAs, yes, you do have multiple copies, but it's not in the hundreds, in the a few copies. Not too bad. Come on.
Hemoglobin?
Same thing. There are multiple copies, but it's not that many.
Transport proteins?
They are in families, but you don't have. One other protein, for example, is histones, right? This whole genome has to be wrapped up around histones. So you need many, many copies of this. And the histone genes are also expressed from clusters. Okay. The difference between gene families and tandem clusters is this. The gene families, the repeated sequences, they can be different. Okay? Alpha and beta tubulin, or these receptors and the transporters, etc., they're gene families, but each gene is encoding for a slightly different protein. In a tandem cluster, the gene is repeated one after another, right? And it's exact same sequence. And that's the difference between tandem clusters and gene families. Does that make sense? No? All right. So non-coding genes, protein-coding genes, together they are about 25%, right? That's not that much. So what the heck is in the rest of our genome? Okay. The rest of our genome contains a lot of repeat sequences. So we have a lot of repeat sequences that are majority of our genome. And now what we're going to talk about is the type of repeat sequences in our genome. All right. The first class of repeat sequences we call simple sequence DNA. So these are usually short tandem repeats. Tandem means right next to each other, right? They are usually 1 to 500 base pairs in size. They're classified further into microsatellites. These are 1 to 13 base pair repeats. You would find less than about 150 of these in one location, in a given location. There are multiple locations, of course. Then there are mini satellites. These are a little bit larger, and you have fewer repeats. And these mini satellites and microsatellites, the different people have different copies of them in their genomes. And because of that, they are used for DNA fingerprinting. Okay? Some of these microsatellites, but not all of them, are actually found near centromeres and near the telomeres. And we can talk more about this as we go into chromosome structure later in the semester. And they have a role in establishing heterochromatin. Again, it's a structure we're going to talk about later. All right. So I said that the copy numbers of these microsatellites is different between me, you, and Geet, and everybody else in this room. Okay? I know him for name because he works in my lab. So I'll embarrass him. But anyway, so this has different numbers. So how do you get different numbers of these repeats in the genome, right? So in normal replication, so these copies of the microsatellites, in a normal replication, so this is your parental strand here, and this is the strand that's being replicated. If everything goes right, you will make exactly the same copy of these repeats, right? This is a 3 base pair repeat, CAG, 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 and everything will be fine. But sometimes DNA polymerase makes a mistake when it encounters repeats. It gets confused, okay? So how does that happen? So here's your parental strand again in green, and it is making the new strand, but sometimes in its catalytic side, DNA polymerase slips backwards, okay? And if it slips backwards, in other sequences, what would happen is that the nucleotides that are incoming would not complement to the parental strand. It would have a problem, it would get stuck, and it would have to resolve this and continue. But when it's in repeats, what happens is it slides backwards, it slips backwards, it can actually just pick up normally, and and starts incorporating new CAGs, right? So this little loop that is newly synthesized just bulges out, but the DNA polymerase just rescues itself and starts replicating normally. So what that does is it adds one more 3 nucleotide repeat to the new strand, right? So when this strand and this strand, they go through a second replication, you will make a daughter DNA with 1 extra repeat coming from this strand, right? And then you will have the normal parental DNA forming the normal daughter DNA. Does this make sense? So this is how the polymerase can add additional copies to these repeats. So if this happens in the germline of a person, then that additional repeat number will be segregated into the progeny. This doesn't occur all the time. It's not very frequent. It's infrequent. And because of that, over millions of years, we end up having different numbers of these repeats, but we can pass that repeat number to our children, right? And then they would have that repeat and pass it, etc., etc. Yeah?
So the backward slippage happens within what? It's what exactly happens with the slippage? It goes backwards in one of the microsatellites, and then it thinks that it didn't count that, so it makes another one?
Yeah, exactly. So it basically, it's going right forward. It slips back a little bit, and bulges what it had made, right? And then it doesn't realize that it had actually replicated that region and replicates it again. Does that make sense in your mind? Can you imagine that? All right. So that's how it does that. So for example, a 3-nucleotide repeat, if it is inside a gene, then it would change the protein sequence, right? So if you have 10 repeats of that 3-nucleotide encoding for an amino acid, you would have 10 of that amino acid. If you have 15, then you would have that 15 of that same amino acid, right? So for example, if this occurs into a transcription factor, and here we have an example of RUNX2 transcription factor. It contains 3 nucleotide repeats in it. And this repeat encodes for glutamine. So this protein has a stretch of glutamine. And the length of that stretch in that protein transcription factor, it alters its activity. And the RUNX2 activity is required for determining the shape of the face. Okay? So it changes how that face has the downward kind of shape. So in this paper, what the researchers have done is they dug out some dog bones from early 1900s to late 1900s. They isolated DNA and then determined the repeat number in RUNX2. And what they found is for certain dogs breeds, what they found is that as the phenotypes gets more dramatic, right? So here is a bull terrier from early 1900s to late 1900s. The repeat number have increased. And the reason why dogs is amazing for genetics is because humans basically cross them to exaggerate phenotypes, right? So they make the poor little chihuahuas and smaller. Pretty soon it's going to fit in my pocket, right? But then it also gives us a very good opportunity to determine the genes that are causing that phenotype. And so in this case, the repeat number in the gene, they had been selecting it for longer and longer repeat numbers, right? All right. So then this shows you that microsatellites and the repeat numbers and the differences in repeat numbers cause differences in phenotypes. And a use of these micro mini satellites in forensics is something called DNA fingerprinting. Okay, so if you watch any of these crime shows, you probably have seen a imaginative gel like this, and so it's basically used for matching the DNA of the suspects to a specimen that's found in the crime scene, etc. Okay, you guys have learned southern blotting lately, right? In the last lecture. Okay, so what's going on here is that they're using southern blots to determine the copy number of a number of different repeats in the genome. So, for example, person 1 has two loci. Let's say this is the first loci and this is the second loci that they're going to look at. And in one of them, it has 9 repeats of a certain mini satellite. And then in another locus, for another repeat, it has, let's say, 8 copies. Okay? And in a second person, for the same loci, right, that person might have 5 copies of this certain repeat, right? And then it could have maybe 13 copies of this one. So at 2 different sites, they have different copies of the repeats. So what the researchers do is they isolate DNA and then they cut the DNA using restriction enzymes outside of these repeats, right? And when they cut the DNA, the length of the repeat containing DNA in this person at this position, is it going to be longer or shorter than this person at this position? If you say longer, raise your right hand. If you say shorter, raise your right hand. Is this going to be longer than this or shorter than this? Longer right, shorter left. This has 9 copies, this has 5 copies. Which one is going to be longer, 9 or 5? 9, right? So this fragment is going to be longer than this fragment. So in a gel, it's going to run higher, right? Does that make sense? So you take that DNA and you run it on a gel. So this is person 1, and that's person 2. This is going to run higher, and in this person, that's going to run lower, right? And for this one, it's going to run, for that person, it's going to run lower, and for this person, it's going to run higher. Make sense? All right. So next step, what they do to visualize these is they use radioactive probes that is specifically recognizes the each of the different repeats, right? And so they mix them up, and so this radioactive probe is going to make sure that these fragments light up into the gel, right? And then this radioactive probe is going to make sure that these fragments are going to light up in the gel, right? And the reason why we have many more bands in a given DNA printing is that they probe many different sites that have contained different repeats, okay? And each person has 2 copies of an allele. Does that make sense? So you have 2 copies of this location. The one that you got from your mom or dad could be 9 copies, but you might get a different one from your other parent, right? And then you would have a different copy number. So what that means is that here it was simplified. So this is the locus for repeat 1, and this is the locus for repeat 2. And I simplified it and said this one has 9 copies and this one has 8 copies. But in reality, what you will have is for each of these, you're going to have a second allele containing a different copy number, right? And this will be your paternal, and let's say this will be your maternal. This will be your paternal, and this will be your maternal. So you can have 9 copies in this, and you can have 10 copies in this, etc. So each locus is going to produce 2 different bands, right? And now you're going to look at many different loci, and together they're going to produce many different bands. Does this make sense? All right. All right, so, for example, in this example, what we're doing is we're doing paternity determination. So we want to know which one of these persons is the father of this child, right? So what you do is you take DNA from the mom, and you determine her DNA fingerprint. You do the same thing for the child and determine her fingerprint. And then you take DNA from father and then also look at that. And then what you do is you try to match the pattern, okay? So, this is the child's repeat number for a given loci, and it seems to match father candidate number 1, right? And it doesn't match father candidate number 2. So already we're kind of starting to say, okay, maybe this person is the father, right? Why does the child don't have this band that you see in the mother? Does anyone have an idea?
Could it be recessive?
Recessive, that's genetics, right? We're not doing genetics. Yeah?
Could it be carried on the X chromosome?
You're getting there, but not quite.
Only half of each
Exactly. So it happened that, so the mother has this band, and then let's say it also has, at the same locus, let's say the mother had 15 repeats of this, and that produced this band. And let's imagine that in the other allele, the mother had 8 repeats, producing this band, right? And when the child is born, it's going to take 1 copy from the mother and then 1 copy from the father, right? So it's going to have a combination pattern, right? And the child happened to take the allele containing the 8 copy, right, and not the 15. So the child does not have this band, but it actually has this band for that given locus. Does that make sense? All right. So the same thing can be done for a specimen taken from a crime scene. And in this case, what you're going to do is you're going to say which of these banding patterns matches which of the suspects, right? And this is not a paternity test, so all of the alleles should be exactly the same. So which of these suspects is actually guilty? 1. I mean, we shouldn't say guilty. Was in the crime scene, right? Let's say that's not the right guy. What kind of twins? Identical twins. So if you have an evil identical twin, you're in really big trouble, right? All right. So that's what it is used for. So the second kind of repeats, we call them interspersed repeats. These are much longer than mini and microsatellites. They can be 100 to thousands of base pairs long, and sometimes even longer. Right? They're found in prokaryotes and eukaryotes, so they're widely found. They're also called mobile elements, jumping DNA, or transposons. They're lost very slowly from the genome because once they get in, they hang out. Okay. So what kind of so this idea of jumping DNA, going from place to place, transposons, was first put out by Barbara McClintock. So she discovered the first mobile elements studying maize in 40s and 50s, but it was very controversial at the time. The scientists were really against this idea that because the DNA carries the information that is necessary to develop an organism, how can you have pieces of DNA jumping around, right? You need some stability to pass the information. So it was very controversial, and she stopped publishing on this subject for a while, but then later her work became recognized when other people start getting convinced by the evidence that there are actually these jumping elements, these mobile elements. All right, so there are 2 kinds of mobile elements. There are 2, I'm going to call them transposons. There are 2 types of transposons. One is called a DNA transposon, and the other one is a retrotransposon. Okay, so the DNA transposons. So here's a figure from your book. The mechanism by which it jumps is a cut and paste mechanism. Okay, so here's your DNA transposon. It is within the donor DNA. Donor DNA is the locus where this DNA transposon is located at originally. So what happens is that this donor DNA gets excised out, right? And this DNA transposon gets excised out from the donor DNA. So this is the former transposon site. So the donor DNA no longer has that transposon. So this DNA, then what it does is it inserts itself into a target site. So it jumps from here to here, right? And it basically cuts and pastes itself. It's jumping from one site to another. The retrotransposons go through a intermediate, and that intermediate is RNA. So the retrotransposon uses a mechanism of jumping that's called copy and paste. So what happens first is that you have this retrotransposon that is inserted in the DNA at the donor site. Then an RNA polymerase transcribes the RNA of this retrotransposon, creating an RNA intermediate. So the donor site still contains a copy of this retrotransposon. Okay, it hasn't left. And then this RNA intermediate is converted into DNA by reverse transcriptase, and then this DNA is inserted into a target site. So now what you have is that you have a new copy of this retrotransposon in the target site and also at the donor site. So it uses a copy and paste mechanism. All right. Yeah?
So they both use the copy and paste, or the cut and paste versus the copy and paste is what makes them different?
Yes. What makes them different is the mechanism by which they jump. Any questions? All right. So here's a more detailed view of what DNA transposons look like. So they contain 2 inverted repeats. These are about 50 base pairs. So there are 2 copies of this. They usually contain the promoter sequences that are required for the transcription of the genes that are encoded by the transposon. So in the middle, you have protein coding region, and it encodes for a protein called transposase. That is the protein that that the transposon uses to transpose itself. All right? So it also, at the target site, where the transposon is inserted, you always see a direct repeat about 5 to 11 base pairs. So if this is ATGCC here, it would be ATGCC here. So it's basically a direct repeat surrounding inverted repeats and surrounding the transposase gene. So in the genome, we have about 3,000 of these DNA transposons. That makes about 3% of our genome. So they're pretty much, there's a lot of them. So these direct repeats is a signature of transposition. And we're going to learn now how that occurs. All right. So what the transposase does for actually removing that DNA is it cuts the donor site right outside of the inverted repeats, right? It makes a very clean cut. Clean cut is a blunt cut. It cuts at the same place in the both strands. Cuts bluntly, right? And then you have a double-stranded DNA with blunt ends. So it removes that. Okay, so you have a blunt-ended double-stranded DNA. At the target site, the transposase does not cut in a blunt fashion. It cuts in a staggered fashion. Okay, so what it does is it cuts the target DNA offset in the 2 strands, right? So what you have is that you have a staggered cut, just like some of the restriction enzyme cuts that you've learned about in the molecular cloning lecture, right? So what you have is that you have a target DNA that contains staggered cuts, a staggered end. And what the transposase does is it ligates the 5' end of the target DNA to the double-stranded transposon in one strand, and it ligates here, okay? And then the cellular enzymes, and sometimes the cellular enzymes recognize that there is a gap in the DNA strand here, right? And it recognizes as a problem. And what it does is it basically adds nucleotides to close this gap and ligates it, right? Just like DNA polymerase would in a replication process, creating this final product, the insertion of the target site. And because it is going to basically add nucleotides that are going to be complementary to this staggered end and nucleotides that are complementary to this staggered end, you're going to have a direct copy of this sequence on the two ends of the DNA transposon, right? And that's how you know that it's a signature of the transposon insertion, because you have direct copies of the two short sequences on both ends of the transposon. And the reason for that is because it cuts it staggered and then fills it up on both ends. Make sense? Okay. So since this is a cut and paste, you're going from one side to another, right? So one of you get up from one chair and move to another. That doesn't create two copies of you, right? No, you just moved. It's cut and paste. But DNA transposons can increase in copy number. Can you guess how this might happen? It's in your book. If you did your reading, you would know this. I see eyes that don't look at me in a very guilty fashion. All right. The way that it can actually increase in copy number is if this cut and paste mechanism occurs during DNA synthesis, in which you have, for example, the DNA replication fork goes in this direction, replicating and making this new DNA molecule and that new DNA molecule, right? It's going in this direction. And let's say that it replicated this DNA, and then this DNA transposon decided to jump from the replicated region of the genome to an unreplicated region, right? And as the replication moves forward, what you're going to do is you're going to make two copies of that DNA transposon so that you have one daughter with a single copy, because this guy jumped from here to here, it's jumped from here to here, but this unfortunate copy is going to have two copies of that transposon because it has now inserted here and then got replicated. Does this make sense? Pretty cool. All right. And if this jumping occurs in the germ cells and gets to the progeny, then that progeny is going to have two copies of that transposon. It's going to pass it to its progeny, which will have two copies of that transposon.
How is the break that happens when the transposon moves from, I guess, the left side to the right side, how is that filled in? Is it just filled in by the same sequence as it was there before, or?
In the previous slide?
No, on that slide. When it moves and then there's that break in the top strand, what happens with that?
Oh, this is what happens there. Oops. This is what happens there, right? So it gets. Oh, come on. Wait. What happens there is in detailed here. In that picture, it's not detailed, so you don't see any staggered cuts or anything like that.
Right. No, I'm not asking what happens on the right side where the transposon gets inserted. I'm talking about the space where it used to be.
Space. Oh, yeah. Now good question. Right. So what happens is that the DNA repair enzymes, which we're going to talk about, recognize that there is a gap. There's a double-strand break. And cells do not tolerate double-strand breaks. Double-strand breaks are the worst thing that can happen to a genome. It breaks it, right? So we have mechanisms that recognize double-strand breaks and immediately ligates it. Okay, so it gets ligated or repaired by homologous recombination. We're going to learn about so that it gets closed. So it doesn't get left out like that. Good question. All right. What about retrotransposons? We have two classes of retrotransposons. One are called the LTR retrotransposons. So these are retrotransposons that have a protein coding regions, and they have these long terminal repeats. They also have that target site insertion direct repeats. They make about 8% of the genome, and these LTR transposons are amazing because they're like retroviruses. So if you look at retrovirus sequences, they are very similar to these LTR retrotransposons. But they don't encode for the genes to make the envelope and the products that help the retroviruses break out of the cell. So you can imagine these guys as the viruses that got stuck in our genome and now live on our genome, right? We have 8% of our genome is viruses. We carry them. So they're stuck. So they're retrovirus-like elements. Another retrotransposon that is very abundant in our genome is called long interspersed elements. These are called lines. There are about 900,000 copies of them, making up 21% of our genome. The full-length lines are about 6 kb long, but many of these thousands of lines are a lot shorter than that. So they are truncated. And then we have short interspersed elements, which are called signs. They make up 13% of our genome, and they're much shorter. And lines and signs, and they're jumping around in our genome, is one of the chief causes of mutations that cause disease, because we have things jumping around, breaking stuff. All right, so these long interspersed elements have an AT-rich region on each end, and it has a protein coding region in the middle. These are called open reading frames 1, open reading frame 2, because they code for 2 proteins. And they jump using the copy-paste mechanism. So here's how they do that. So pay attention to this. And these kind of diagrams in your book, the best way to learn these is to just try and draw it yourself. Because if you actually try and can replicate it, then that means you actually learn the process. Okay? So what happens is that this ORF2 protein, so the first thing that happens is the retrotransposon gets transcribed, right? And at the end of transcription, it gets polyadenylated, just like any old mRNA. And it has a number of A's at the end, right? So the ORF2 protein takes that RNA, the line RNA, and goes to the target site and matches a stretch of T's to the A's and creates a cut that is again staggered, so it makes nicks on 2 ends. And what it does is it takes that DNA strand, pulls it over, and now uses the 3' end of that as a primer to copy the RNA sequence into DNA. Does that make sense? It's grabbing the 3' end of its target DNA and started to use it as a primer to extend. So it's going to extend and make the DNA further and further, and as it extends, it's going to make a copy of the RNA, and at the end, it's going to reach the end of the strand that it is using as a primer. Okay, so it basically made a DNA copy of the line RNA. So you have a DNA-RNA hybrid in the target site. So what kind of enzyme is ORF2? It's making a DNA copy of an RNA. You guys learned this? Reverse transcriptase. Very good. Nice recall. All right. So you have this DNA-RNA copy. Again, the cellular enzymes recognize this as a problem. And whenever they see this, they say, okay, we're going to repair this. And the way that they do that is they remove this RNA, the line RNA, and use the 3' end of the other strand as a primer to extend and replicate so that it reaches the end and it gets ligated. And then what you have now is you have the line DNA inserted at the target site surrounded by 2 copies of the insertion site, the 2 direct copies of short insertion site that was created due to the staggered cuts. All right, so that's how that happens. And here's the picture again. What you have is you have the staggered cut, and the ORF2 grabs into this end, the 3' end of one of the strand, and uses as a primer to start copying the line RNA. And it goes on until it reaches the end, usually. But when we look at in our genome, we find a lot of line elements that are truncated. And they're truncated at this end. So they're truncated at the 5' end. Okay? So what do you think? What property of ORF2, in general reverse transcriptases, could lead to this? Lead to this truncation at one end?
They generally tend to fall off before they finish transcription.
Very good. These enzymes have low processivity. That means how long can you hold on to your template? How long can you incorporate nucleotides and go along? And because they have long processivity, they fall off, right? So this guy is making this, making a copy, and if it falls off before it reaches the end, like this end of the line RNA, what you're going to have is you're going to have a line element that is essentially that long, right? Instead of the whole thing. So that's how you have lots of truncated copies of the line elements. And what would be the consequence of having a truncation in your line insertion? Would that new line element that's truncated, will it be able to autonomously, what that means, by itself, able to jump again? Yes or no? It's not going to be able to jump again. Why?
Because it won't be able to code for the necessary enzymes.
Yeah, you have truncated your ORF2 or some other enzyme that is necessary, and then you won't be able to jump by itself. But will it be able to jump using the machinery from a full-length line element? In some cases, yes. Only if it's able to get transcribed, right? If you have a line RNA again that the ORF2 from another line can grab, then it will be able to get inserted. So it's not autonomous anymore, right? It depends on somebody else to get inserted, but it will be able to get inserted. All right. So that brings me to signs. These are short interspersed elements. They're about 100 and 400 base pair in length. They make up 13% of our genome. In humans, 70% of these signs are called allele elements. The reason why they got that name is because they contain a restriction enzyme site for allele. So these repetitive sequences contain a restriction site for allele 1, and that's why they're called allele elements. So these short allele sequences are transcribed by RNA polymerase III, and they're also partial allele sequences. Just like the lines, they tend to get truncated. These sign elements are not autonomous. They're too short. They don't encode for transposases, so they can't jump by themselves. But what they do is they can use the transposition proteins encoded from the lines, so they can be grabbed by ORF2 and then jump non-autonomously, right? So they can jump like truncated lines. All right. So the transposons jumping around, the one consequence of that is mutations, right? Imagine a line that by mistake jumped in the middle of a very essential protein that's going to cause problems, right? But also there are other consequences to transposon jumping. For example, transposon jumping can make pseudogenes. Pseudogenes are genes, when you look at their sequences, they are very similar to the sequence of a gene that's already present, but it has accumulated lots and lots of mutations, okay? So, and it doesn't encode for a protein product anymore. So it's called a pseudogene. Looks like a gene, but it's not. So let's say you looked into your genome and you found a region that has its DNA sequences very similar to a gene called translated region gene, okay? Very similar, but they have lots of mutations. So they're kind of all degenerated and it doesn't encode for the protein anymore. It accumulated lots of stop codons, but it's there. And the next thing that you recognize is, wait a minute, this translated region is surrounded by 2 short direct repeats. That tells me that this pseudogene has come to existence because of a transposition event. Okay? And we know there are 2 kinds of transposition events. One is cut and paste. The other one is copy and paste. Which one do you think this transposition used? Cut and paste or copy and paste? Cut and paste? Why do you think that?
Because the records are just separated, they were like replicated.
It has these short direct repeats.
So that would be copy.
Well, both cut and paste and copy and paste causes short direct repeats. So that's not a distinguishing feature. Think a little harder. What's the big difference between cut and paste and copy and paste? Tell me.
The copy and paste uses RNA.
Very good. It has an RNA intermediate. It's RNA that's polyadenylated process just like mRNAs. So what will happen? What kind of intermediate is that now? Will it have introns? No, right? Do you see any intron sequences in this? No. So then you would predict that this was made through a copy and paste mechanism, right? And in fact, the ORF2 protein is supposed to grab the line RNAs for transposition, right? But by mistake, sometimes it can grab onto a normal mRNA that has a poly A tail and accidentally insert a copy of that inside the genome, right? Very good. There are other things that transposons can do that are amazing to our genome. One is called exon shuffling. What it can do, what transposon insertion can do, is it can take an exon and then put it inside another gene and create another exon for that gene, right? So imagine a protein, let's say it has several different domains, and let's say one domain, like a DNA binding domain, was encoded from one exon. And if that domain, by chance, is taken by transposition and inserted into another protein, then that protein is going to have the DNA binding domain of the original gene, right? And that's something that happens, and it's called exon shuffling. And it can be done by both DNA transposons and retrotransposons. For example, the DNA transposons, if there are if it happens that there are 2 DNA transposons surrounding a exon, right? And the transposase accidentally, instead of cutting here and here, right, it cuts here and here. What it will do is it will excise that whole DNA fragment containing the 2 transposons and the exon in between them. And the transposase enzyme actually doesn't know any better. What it will do is it will take that DNA and insert it into a new site. So, and if it happens to be inside a gene that has a very long intron, and we know the genes have contained lots of long introns, then you basically shuffled in a new exon, right? So this can happen also by retrotransposition. Again, imagine a gene where you have a line element inserted here. It has a weak poly A signal usually, and then the gene has a stronger poly A signal at the end of its last exon. Imagine that you have the transcription of the line starts right here through its promoter, but the transcription accidentally continues into the last exon and gets polyadenylated at the end. You will create an RNA intermediate product that's going to contain the line and also the exon, right? And if the transposition event now inserts it into a second site, what you're going to have is you're going to have a copy of the line, and it also brought in, along with it, the last exon of the original gene, right? So that's how it can shuffle in a new exon and create a new gene. All right. So with the repeats, which are the majority of our genomes, we cover about 75%. The remaining 25%, 20% of the genome is unclassified. And these regions of the genome usually contains a lot of regulatory elements and other things that we don't know what they do, right? So nowadays researchers try and figure out what they do. So these are called unclassified spacer DNA. All right. In our cells, we have what we talked about was our nuclear genome. So that's the genome inside our nucleus with all the chromosomes. But we also have what is called the mitochondrial genome. And each of us have inherited our mitochondria only from our mothers, right? So there's something that your dad can never do for you, is contribute mitochondria, right? We only have mitochondria from our mothers, and that mitochondrial genome is the same genome of the mitochondria that's coming from your mom. So the mitochondrial genome encodes for genes that are mostly used in the mitochondria itself. And in plants, you also have the chloroplast genome, right? And that's the genome that's used in the chloroplasts. So these are non-nuclear genomes for organisms. And in humans, for all these mitochondria functions and all these proteins and RNAs that are used in the mitochondria, some of these products are encoded from the mitochondrial genome, but some of them are encoded from the nuclear genome and then gets transported inside the mitochondria. So the mitochondria is made in a collaboration between the mitochondrial genome and the nuclear genome. All right, so I usually put some animations if you guys want to look at it to learn some of the subjects. And then I have lots of study questions, so go ahead and study that so you can keep up with the coursework. All right, see you next Monday.